who you are, and then.
So I've just started the recording.
Okay, so good afternoon, everyone. We have here Dr. Kelly Speer now from the Smithsonian Institution. Kelly is a Biodiversity Genomics Postdoc at the Smithsonian National Museum of Natural History and Conservation Biology Institute. She received her Ph.D. from the Richard Gilder Graduate School, the degree-granting institution with the American Museum of Natural History, and her master's from the University of Florida and Florida Museum of Natural History. Kelly studies the interactions between hosts, parasites, and microorganisms to more accurately assess the response of complex communities to stressors like climate change, habitat loss, and novel disease emergence. I'd like to welcome you all to Dr. Speer talk, which is going to be how host-parasite-microbe interactions inform animal ecology. With you, Dr. Kelly Speer.
Thank you very much for having me, Hernani. I'm sorry for the confusion about the time difference again, but I'm looking forward to talking to you all about some of the research I've done. So let me just share my screen. Are you all seeing my presenter view or like the slides?
We can see the slides. Yeah.
Okay. Okay, great. So my research leverages knowledge about hosts, their parasites, and their microbiomes to examine complex hypotheses about how communities respond to environmental change like climate change, habitat loss, or novel disease emergence. Generally speaking, when you're talking about the microbiome, the old adage is that everything is everywhere, but the environment selects. This has sort of been, I guess this was the first attempt at characterizing microbial community ecology or microbial biogeography. And we have since learned that, okay, bacteria are not everywhere that we expect them to be with only the environment determining, but it's a useful null hypothesis for us to test more realistic systems. So keep this in mind as I go through my talk. So the system that I particularly work on are bats and their blood-feeding parasites called bat flies. So bat flies only feed on the blood of bats. They also vector some pathogens to their host bats, for example, malaria specific to bats and some bacterial and viral pathogens. And their microbiome communities, the microbiome communities within bat flies, impacts their ability to transmit those pathogens. Here are where the bat fly families fall out phylogenetically. So there are two families of bat flies, the Nycterabiidae and the Streblidae. They are sister to the Hippobosidae, which are head flies that feed on birds, mammals, some feed on reptiles, and the Glossinidae, which are tsetse flies and vector important human pathogens. And all of these families of flies belong to the Hippoboscoidea that are linked by having a really interesting life history strategy where the female flies actually develop their young internally. And these fly larvae hatch inside of the female and then feed on milk glands until they're ready to be deposited in the environment. And then after they're deposited, they emerge from a puparium, which is sort of like a cocoon for a caterpillar, and then they turn into adult flies. So just so you know, I'm going to be using these two silhouettes throughout the talk to describe the Nycterabiidae and Streblidae, these two families of bat flies. Here, just for some broader context, here's the group of Diptera that they fall within. They're pretty closely related to Drosophila, actually, fruit flies, which is interesting. So what I do mainly with my research is I characterize the microbiome of these bat flies. So how do I do that? Well, I do a lot of field work, and I also rely on a lot of collaborators, including Brazilian collaborators like Hernani, to collect bat flies. So here's just a little video of how we collect bats. A lot of you might be very familiar with this already, but essentially we set up nets in the fields and capture these bats in the nets, and then take them out of the net, and take them back to a field lab where we work on them. So this is a time-lapse video, which actually I think just got paused. I'm not sure if you all can still see it. It's a time-lapse video of a field lab that we have in Belize, where there's actually over 50 people doing different types of research, all of those bags hanging on the line. Each white bag contains a bat, and everybody going up to the line tying flagging tape on those bags is someone interested in a different aspect of that bat. So some people are doing disease ecology, sampling the saliva and fecal matter of those bats. Other people are doing morphology, looking at how like the muscles in the wings connect and allow bat flight. I'm looking at the parasites in their microbiomes. So here's a video of how I use forceps to pick little bat flies off of the wing of bats. This is a very pregnant bat. That's why its belly is so round. So I've essentially just gotten very good with a pair of tweezers. Okay, so once I've done the field work or relied on collaborators to get those samples for me, I take those bat flies back to the lab and I do DNA extraction and library preparation and then sequencing. For most of the microbiome work I do, I use metabarcoding, which relies on a single locus that you universally amplify within a sample, and it tells you something about the composition of that sample. So many people do metabarcoding if they have like thousands of insects in one sample that they want to just figure out what the taxonomic distribution of that sample is. So they use a barcoding locus to identify all of the insects in that sample. I use the locus, the genetic locus 16S rRNA, which is the primers that I use are specifically targeted to bacteria. So whenever I do my library preparation, I'm actually amplifying this one locus from all of the bacteria present in my sample. So all of the bacteria present in a bat fly. And then I use Illumina MySeq to sequence, hundreds of bat fly samples on a single run of sequencing, and I get all these reads out at the end. So why is this important? Why should we care about bat fly microbiomes? Well, insects with narrow diets, like parasites, have really interesting microbiomes. These microbiome communities are dominated by endosymbiotic bacteria. So these are bacteria that have evolutionary relationships with their hosts. And these endosymbiotic bacteria are maternally inherited. They're encased within a specialized organ in their host, and they provision nutrients missing from their host diet. So in the case of parasites that feed on blood, blood meal is actually really low in B vitamins, which these parasites need to survive and reproduce. So it's hypothesized that the bacteria actually produce those B vitamins and give them to their host, creating this beneficial symbiotic trade-off between the insect and the bacteria. There are also bacteria that are facultatively associated. So these ones aren't maternally inherited. They're sort of just acquired from the environment. They're less strictly associated with their insect host. These also can supplement nutrients not provided by the endosymbionts. They can contribute to host immune response upon pathogen invasion. So if you remember back to that tsetse fly I was telling you about, we know that the bacterial community in the gut of tsetse flies helps the tsetse fly form a physical barrier to the invasion of some pathogens. And so it makes the tsetse fly better or worse at transmitting pathogens depending on what gut bacteria are present in the fly. These facultative bacteria also generally form a more depauperate community than we see in vertebrates. So you might remember Melissa Ingala's talk when she was talking about bacteria associated with bats, and she probably showed you plots where there were like hundreds, if not thousands, of bacterial taxa. For these insects, we're talking about tens of bacterial taxa. The community is really depauperate. So while the endosymbionts, these endosymbiotic bacteria are maternally inherited, what is the source of the broader microbiome community in this system of bats, ectoparasites, and microbiomes? One hypothesis is that the microbiome is colonized by a subset of environmental bacteria. Another hypothesis is that the microbiome more members of the microbiome may be vertically or horizontally transmitted, so may be inherited from mother to offspring or even inherited from other members of the same population. And the third hypothesis is that it could be both the environment and the insect community determining the microbiome composition. And this all has impacts for our understanding of disease transmission if the microbiome impacts vector competence. So let's look at the first idea. How does environment impact microbiome composition? So in our bat fly microbiome setup, this is essentially looking at just this arrow here, just the environment going to bacteria and then to the bat fly. One valuable null hypothesis that we can use is island biogeography, which says that if we treat these little gray circles as patches of habitat or islands separated by ocean, island biogeography tells us that the community of organisms on small isolated islands should be much less diverse than islands that are closer to a source or the mainland. So using this as our null hypothesis, we can start to ask, okay, how does habitat fragmentation impact the composition of microbiome communities in bat flies? And for this, I actually relied on one of my Brazilian collaborators, Thiago Teixeira, which I'm sorry if I brutalized that name, please forgive me. But he did a lot of sampling for his PhD in the Atlantic forest of Brazil. And he sampled from all of these different fragments. So the large green area that's outlined in white at the top of the slide is actually a remnant protected reserve called Regua. And he sampled 3 times within Regua. And then outside of Regua, he sampled 10 additional habitat fragments, patches of forest separated by agricultural land. From these sites, sorry, let me back up. So this is really good because all of these different forest fragments that he sampled from vary in their shape, their isolation, and their area. So it allows us to apply this island biogeography null hypothesis to studying microbiome composition. So he sampled these different bat species that feed on fruit, blood, and insects, and these different bat fly species. So remember, this is the Nectar silhouette here on the left, and these are the Nectar species. And here are the Streblidae, the other family of bat flies, and the species that we sampled from this family. Okay, so let me show you some microbiome plots. So this is just a qualitative look at the microbiome composition in different bat fly species. So each bar here represents a different bat fly species. The colors in the bars represent the different bacterial genera that we detected in each of those species. So I've outlined the Nectaribiidae family in black and put the silhouette below those bars for you. What you'll notice right away is that while the Streblidae are completely dominated by Arsenophilus, this yellow color, that bacteria seems to be completely absent or really low in relative abundance in the Nectaribiid species. Arsenophilus is the hypothesized primary endosymbiont in bat flies, so it's expected that it's provisioning B vitamins that the bat flies need to survive. So it's really weird that we don't see it dominating the Nectaribiid species in this system. Instead, the Nectaribiid species are really dominated by Wolbachia, which is a bacteria that's known to cause mitochondrial DNA sweeps because it can actually be a reproductive parasite of insects. In some cases, it may facultatively provision nutrients, but that beneficial relationship is really rare with Wolbachia. The other genus that's really dominant in Nectaribiid bat flies is Bartonella. Bartonella is a bacterial genus that contains some human pathogens. For example, Bartonella quintana causes trench fever in humans, and it's hypothesized to be an arthropod vectored pathogen of mammals. So previous research has actually hypothesized that bat flies are the primary vector of Bartonella in bats. And we might be finding here that it seems like one family is much more susceptible to Bartonella than the other. So let's take a more quantitative look at what our microbiomes look like. This is a principal coordinates plot. So each of these dots here represent a different microbiome community. And when 2 dots are close together, that means that they have similar microbiome compositions. And when two dots are far apart from each other, that means they have different microbiome compositions. The different colors here represent the different bat fly species. You can ignore the shapes. So all of the darker, like purpley blue colors, those are the nectar bead bat flies, and those are distinct in this plot from our strepsillid bat flies. And when we try and see how well parasite species is able to explain microbiome variation, we find a significant explanatory power down here from Pervanova. Okay, so how does being within regua, or essentially island size, impact microbiome composition? So here the circles represent all of the samples that were collected outside of regua, and the triangles represent all of the samples that were collected within regua. There's not an obvious pattern here when you're just looking at it, like kind of squinting your eyes. The main pattern seems to be, again, that bat fly species is a strong explanatory variable for microbiome composition. So let's break this down more clearly based on island area and isolation. So here I'm showing you plots of bacterial taxon richness on the y-axis of both plots by sampling site. And in the plot on the left, the sample sites are ordered by decreasing area. And on the plot on the right, they're ordered by increasing isolation. So if island biogeography was explaining the microbiome composition at these sites, we would expect to see a downward trend in both plots, but we don't see any trend at all. There's no significant difference between bacterial taxon richness, which here is labeled ASV richness. And if you want to talk more about what that means later, feel free to ask me. But bacterial taxon richness does not appear to change based on where a bat fly was sampled. But bacterial richness is only 1/2 of microbiome composition. Remember that the relative abundance of bacteria in the microbiome can also change, and those changes can have a big impact on how the microbe functions within that community. So in order to assess how relative abundance of bacteria changed between samples, we used ecological interaction networks. So here, let me just walk you through how to read these plots, because I know they're kind of weird. Each dot in this network represents a different bacterial taxon. And when you see a gray line connecting two dots, that means that the relative abundance of those two bacteria is correlated with each other between samples. So it's hypothesized that that is a proxy for interaction between those two bacteria. So here are 3 networks from the 3 sites that Tiago sampled from within regua. You'll notice that all of the dots, all of the bacteria are really well connected. There aren't very many bacteria that have really big impact on the network, and there are no really isolated pockets of neighborhoods of bacteria. And here are 2 examples of networks from our smaller fragments. They look much different than the networks that we see within regua. Instead of having well-connected neighborhoods across the network, we see isolated neighborhoods of interacting bacteria, and we also don't see a big importance of arsenophilus anymore in holding the network together. But for those of you, and this is a significant difference. So modularity is a measure of interaction network. It's like the degree of separated neighborhoods within an interaction network. And so there's a significant difference between networks constructed from samples outside of the reserve and samples collected within the reserve. But for those of you who are really eagle-eyed, you'll notice that our sample sizes are really different for building these networks. The ends at the bottom left of these plots indicate the sample sizes used to reconstruct the network, and the M is the number of parasite species represented in the network. So generally speaking, the regua sites support a bigger and more diverse parasite community than the smaller sites do. So this could indicate that habitat fragmentation has trickle-down effects through a community. So by decreasing the bat species that can occupy a site, it decreases the parasite species that can occupy a site, and then has subsequent consequences for the microbiome community. But what if we control for this difference in network size? Do we also still detect a direct influence of habitat fragmentation on network structure? So this plot is like a little confusing, so just bear with me. The answer is yes. Habitat fragmentation does have a conserved change or have a conserved impact on microbiome community structure. So here, each of these dots actually represents a bacterial network. When two dots are close together, it means that the structure of those two networks are more similar to each other. When they're far apart, it means that the structure of the underlying networks is very different. The dark green dots are networks that were reconstructed from samples within regua. The light green dots are networks reconstructed from samples outside of the reserve. And when you see gray lines connecting two dots, that indicates that they were reconstructed from the same parasite species. So take, for example, this dark green dot at the top left of the plot next to Aspidoptera falcata. So Aspidoptera falcata is the name of a bat fly species. This dot represents the network that was reconstructed from this species within regua. And then if we follow this gray line down to the light green dot, this is the network that was reconstructed from the same bat fly species, but outside of regua. And we see this conserved change between within regua networks and outside of regua networks in all of the species we had enough samples to test, with the exception of this species here, Basilia juquiensis. And this is the only Nycterobiid species we had enough samples to do this for. So the difference that we see in this regua network compared to the others is likely just due to the differences between Nycterobiidae and Strepilidae, rather than a violation of the pattern that we're seeing. So essentially the take-home message from this plot is that bacterial networks respond to habitat fragmentation both directly and indirectly filtered through changes to the parasite community. So from this research, we know that microbiomes are very host fly specific. The specificity of the microbiome to the fly may mediate the impact of habitat fragmentation on diversity, and habitat fragment area changed bacterial network size and shape. So we know now that the environment has direct and indirect impacts on the bat fly microbiome. So how does this compare with the contribution of the bat skin, for example, which can also be treated like the environment when we're thinking about a parasite, right? So what are the relative contributions of host and environment? So for this project, I sampled bat flies from this bat, Leptonicteris yerba-buena. It's a pollen and nectar feeding bat from Mexico and Central America. I don't think it actually gets down to Brazil, but maybe I'm wrong. Okay. So this is, yeah, thanks for helping me, Hernani. So yeah, here's another, this bat pollinates like cacti, and it also pollinates agave. So if you like tequila, then this bat is like, gotta be on your top 10 favorite list. Here's another picture of it just being really cute with pollen all over its face. So it's really good at its job. So the nice thing about this bat is that it's migratory. It spends the fall and the winter in central and southern Mexico, and then it migrates annually and spends the spring and summer in Baja and northern Mexico. So this allows us to test the hypothesis that geography, so the broader environment, or the bat skin is impacting bat fly microbiomes in the presence of high dispersal. So we're essentially eliminating the impacts that dispersal might have in changing the microbiome communities between these sites. So the way that we did this, our experimental design, is that we, from these 5 different cave localities, I swabbed the inside of the cave 5 times, and then I sampled bats from that cave, and I swabbed the skin of those bats, and then I collected their ectoparasite species off of them. And I'm going to introduce this new silhouette here. So both of these bat fly species are streblets, but this is going to represent Trichobius sphaernotus, which don't worry about the name, I'll tell you again later. And then this is going to represent Nycterophilia coxata. The reason this species doesn't even look like a fly is because it's laterally compressed like a flea, and you can't even put it on its stomach because it's so skinny. You have to essentially always lay it on its side. So that's why it looks like that. For the rest of the talk, I'll be referring to these collection localities as Northern Baja, Southern Baja, and the mainland. So here's our qualitative look again at the composition of the microbiome in these different sample types. This little trapezoid thing represents our caves. Then we have a bat silhouette, our batfly species 1 silhouette, and our second batfly species silhouette. So there seems to be right away a stark difference between the environment, the bat skin and the cave microbiomes, and the batfly microbiomes. With our Trichobius sphaernotus species, this species here, being dominated by Arsenophilus, and the Nycterophilia species being dominated by a Nycterophilia-specific endosymbiont. This has previously been described. It's a unique bacteria to this genus of bat flies, and it fills the same role that Arsenophilus does for other bat fly species. So it provisions missing B vitamins from the diet. One interesting thing to note is that I've again outlined Bartonella and Wolbachia for you all, and it seems like there's a much higher relative abundance of Bartonella and Wolbachia in Trichobius sphaernotus than there is in Nycterophilia coxata. So let's look at how geography impacts these samples. Just looking at the cave and bat first, when we look at our Northern Baja cave, sorry, let me orient you to this panel first. So here, each of our bars represents a different sampling locality. The colors in the bars still represent bacterial taxa, but then I've broken apart these bars into different panels based on their sample type. So just remember that, so here we have the cave samples, and then each of these bars represent a different sampling site and the microbiome composition from that site. So within the cave samples, when we look at our Northern Baja site and then compare it to our bat skin samples, also the Northern Baja site, we see that their microbiome communities look pretty similar. And we see that across all of our sampling localities, and that there is a lot of geographic variation in the bat skin microbiome according to cave, despite high dispersal between these sites. We don't see very much geographic variation in the bat fly microbiomes. Instead, it looks pretty consistent across all of our sampling localities. When we use principal coordinates analysis again to examine the differentiation between microbiome communities by sample type. So here, each of the colors represents a different sample type, and the circles represent samples that were collected from Baja, and the triangles represent samples that were collected from the mainland. But the big take-home message here is that the batfly microbiomes indicated in black and purple are distinct from each other and also distinct from both the bat skin and cave, where the bat skin and cave microbiomes are almost completely overlapping. So they're hardly different at all. Sample source is a significant determinant of microbiome variation according to Permanova, but one caveat to Permanova is that if you have different sample sizes between your groups, then the homogeneity of variance, so like essentially the amount of variance that is occupied by one type of sample has to be similar between groups. But unfortunately, my samples violate this assumption. So it's hard to believe Permanova is a good tool for assessing the system. In order to look at how much signal there is in the data for sample type, we used classification by random forests. So random forests are a machine learning algorithm that are really good for categorical data. They don't make underlying assumptions about the distribution of the data. And while they still suffer when sample sizes are uneven, you can use resampling to fix those issues. So here we used a variable of interest as the outcome and the microbiome as the predictor variable of interest. And so our equation looked like this, where sample type was the outcome we were trying to predict, and the information that we provided the model was the microbiome variation represented by principal coordinates axis 1 and 2, and then also collection locality. When we do this and see how good our model does at classifying a subset of our data, we find that it does significantly better than a no information rate, essentially significantly better than random. And the only times it ever misclassifies a sample to a sample source is between bat skin and cave microbiomes. It never misclassifies the bat fly species as each other or as their environment. So this gives us good confidence that microbiome community variation is very strongly explained by sample type. But remember that I told you that the bat fly microbiomes are almost completely dominated by their bacterial symbionts. So what happens when we remove those? So just in silico, using my computer, I removed every single read belonging to either Arsenophilus or the Nyctrophilia specific endosymbiont. And I reordinated the data to see if we still see distinction between our samples. So even though there seems to be some overlap between the Nyctrophilia coxata microbiomes and the environment, our random forest model is still able to classify these bat fly microbiomes with 100% accuracy as distinct from the cave and bat skin, even when we remove their bacterial symbionts. So that indicates that even the facultatively associated bacteria are distinct from the environment. So here I'm just looking at bat skin and cave to get a better idea of what's going on, because it seems surprising that the bat skin microbiome would vary so strongly by geography, especially given these bats are annually moving between these cave sites. So here, the different colors represent our different sampling sites, and the triangles represent our cave samples at each of these sites, and the circles represent the bat skin microbiomes at each of these sites. So what you'll notice is that all of the samples collected from the same site, so of the same color, cluster together very closely, indicating that geography is a strong explanatory variable of bat skin microbiome here. And this sort of contradicts what people have previously found for human microbiomes. They found that humans generally alter the microbiome of their environment, but here it appears that actually the cave is altering the skin microbiome of the bats very rapidly. So bat fly microbiomes are distinct from their environment and the bat. There is strong geographic variation in bat skin and cave microbiomes despite high dispersal. And there's potentially differential susceptibility between bat fly species to Bartonella and Wolbachia, which sort of supports what I showed you in the first part. So, okay, now that we know that the environment impacts the bat fly microbiome, but that there's still aspects of the microbiome that are distinct, that may be more vertically or horizontally transmitted, indicated by this arrow. What are the consequences of this variation for pathogen transmission to their bat hosts? So to examine this, I have been doing a multi-year survey in Belize, a Central American country. I've sampled bats and their bat flies from these two different sites. So this checkered outline site at the bottom of your screen is actually a protected reserve that's called Laminae. And then the site that's outlined in black at the top of your screen is a fragment of forest that's separated by agricultural land. And the reason it hasn't been deforested is because it actually contains unexcavated Mayan temples that serve as really good bat roosts for many bat species. Just to tell you what the other colors on this plot are. So the white is land that was cleared before the year 2000, and brown is land that was cleared between 2000 and 2014. So as Kakabish, this fragment of forest at the top, becomes more and more isolated from other forest fragments nearby, we want to monitor how the consequences of this for bat fly microbiomes and pathogen transmission in bats. So far, this is part of an ongoing study. Excuse me. I've sequenced the microbiomes from 182 bat flies, and we've screened over 700 bats for Bartonella, that bacterial pathogen I mentioned, and also Polychromophilus, which is a bat-specific malaria. So these are the bat species that we have looked at so far. And again, the bat fly species broken apart into the Nyctorobiidae and Streplidae. When we look at microbiome variation by bat fly species indicated by the different colors, we again see a strong explanatory power of parasite species in explaining bat fly microbiome variation. When we look, I just want to show you 2 years that we've sampled. So each bar here is a different bat fly species, and again, the bacteria are the different colors. And then the panels just represent 2 different years that we've sampled. And the Nectar-Biidae are currently outlined in black. So remember back that I told you that Brazilian Nectar-Biidae don't have Arsenophilus. Well, Belize Nectar-Biidae do. So there seems to be some really weird geographic variation going on in this family in the neotropics, especially in Brazil. So if anyone is interested in bat fly microbiomes, there's a lot to do in Brazil. So then let's take a look at the Streptilidae. Again, we see that most of them are dominated by Arsenophilus, totally expected, except here's another little wrench. These 2 species are dominated by the Nycterophilia specific endosymbiont I mentioned a couple minutes ago. This is expected for this species here. Can you see my cursor? Yeah, okay. So this species here, Nycterophilia parnelli, it's a member of the genus Nycterophilia, so it's not surprising that it has the Nycterophilia specific endosymbiont. But this species is Trichobius younkeri, and every other member of the genus Trichobius has Arsenophilus. This is the only time that any other species outside of Nycterophilia has been found to have the Nycterophilia specific endosymbiont. And to just make this a little weirder, these 2 bat fly species are specific to the same bat species, Pteronodus mesoamericanus, this bat here. So what I did is I just ordinated the microbiomes from these 2 bat fly species based on whether or not they were male or female. So here, Trichobius younkeri is blue, and Nycterophilia parnelli is red. And the females are circles and the males are triangles. So not only does Trichobius younkeri have a symbiont it's not supposed to, it seems like the males are undergoing this transition. The males of Trichobius younkeri are closer in their microbiome composition to Pteronodus mesoamericanus than the females of the same species. So this is a really weird thing that I really don't understand what's going on here yet. It could be that we're witnessing a transition where the Nycterophilia specific endosymbiont is outcompeting Arsenophilus and is a better symbiont or provides better nutrition. But that doesn't explain why it seems to be sex biased. So very weird, interesting stuff. Still working through that. It's like a good brain teaser. And if you guys have any good ideas, I'd love to hear them. Okay, so let me just talk to you a little bit about what signal of habitat fragmentation we've been picking up in the microbiome so far based on our first 2 years. So here I've split it just based on the bat fly species that we have sampled between our fragmented site, Kakabiche, and our reserve site, Laminae. And I've split it up also by sampling year. So here's what our Kakabiche microbiomes look like between 2017 and 2018. There's some variation occurring between years, particularly in Trichobius younkeri. And then when we add in the Laminae information, it gets a little more interesting because suddenly we see a much higher relative abundance of Bartonella in Laminae, which is our protected area, than we see in Kakabiche. Now, this is contrary to what our baseline hypothesis is, that larger, healthier environments lead to lower relative abundance of pathogens. But in the case of Bartonella, it seems like Laminae, our protected site, has higher relative abundance of this pathogen, at least in the bat flies. So this is still ongoing. It could just be a 2-year blip that we captured. Perhaps there was like a heavier rain year at Laminae than in Kakabiche. So there are other explanations for this that we're continuing to monitor. So let me tell you what the results of our pathogen screening was in bat blood. So Bartonella is extremely common in Belize. Over 77% of the individuals we screened were positive for this bacteria. However, bat-specific malaria, polychromophilus, was extremely rare. Only 5 individuals of the over 700 we screened tested positive for polychromophilus, but we did detect it in a new genus of bats that it's never been detected before in, Rogisa, an insect-eating bat. Interestingly, polychromophilus is very rare in the neotropics, but it's extremely common in Africa, Europe, and Asia. It is often found at prevalences of over 50% in European bats. So it's really interesting that it's so rare in the neotropics. There was no significant correlation with bat fly microbiomes and the prevalences of these two pathogens. However, we know from previous research that the interactions between microbiomes and pathogens is normally very strain specific. So by looking at pathogen strain, we might get better resolution for this question. So bat fly microbiomes vary by year and sampling site. Bat fly species is the strongest predictor of microbiome composition. And there's no evidence that bat fly microbiomes drive prevalence of Bartonella and polychromophilus yet. So taking this holistic look, examining the environment, the bats, the parasites, and their microbiomes gives us a better idea of how complex interactions respond to environmental change. And as we continue to see destabilization of communities and extinction of species in response to climate change or habitat loss, it's going to be more valuable than ever that we take this sort of holistic view. For my postdoc, just to give you a little bit idea of where I'm going with this research next, I'm actually looking at the microbiomes of bat ticks, which are also important vectors of bat pathogens. I'm trying to see how their bacteria vary from bat flies feeding on the same bat individuals. So what is the phylogenetic association between a parasite and its microbiome? I'm also interested in looking at the evolutionary transitions to blood feeding in insects. So here is a plot of different insect families that are closely related to bat flies. So these stars are bat flies plus the ked flies. So blood feeding has arisen a couple times in this group of flies, once at this superfamily that includes bat flies, and then again here in the Mucidae. So what is the role of bacterial symbionts in enabling these transitions to blood feeding and promoting essentially vector competence? So I've been using historical specimens from the Natural History Museum in Washington, D.C. to address this question. And I've generated some results from doing metagenome sequencing from these samples. So here, this is what's called a Busco assessment, and it tells you essentially how good your sequencing in assembly is. So here we have different, each bar represents a different species that I've sequenced. The blue are the genes that we've captured in their completeness. The yellow are the ones that we've only captured part of, and the red are ones that are totally missing. So for the most part, we do a very good job when doing metagenome sequencing at getting good assemblies for flies, with the exception of this fly, which is the tsetse fly, and it has a huge genome. So it's probably an issue of sequencing depth. But it seems like metagenomics is a good option for these flies, even in historical specimens. So yeah, if you want to talk about that, we can also talk about that. But thank you very much for your time. I really enjoyed talking to you, and I'm looking forward to our discussion. I can take any questions.
Okay, thank you, Kelly, for for giving the talk as well and taking your time. If you want, you can stop recording, I think, right now, like, because usually I uploading the talks, but not the conversation. But it's up to you as well.
Sure. Yeah. Let me figure out how to do that. I might have to share my screen first. Okay. Yes. Stop.